Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're investigating the effect of temperature on initial rate of an enzyme catalyzed reaction. And this also includes talking about the temperature coefficient, which is Q10. So an enzyme-controlled reaction is influenced by various factors, and these factors are temperature, the pH, the concentration of the substrate, and the concentration of the enzyme. And if you want a more detailed video on enzyme-controlled reaction, like the actual theory, video's already been posted, so do check that out. This video will only be focusing on the practical side of things. So the initial rate of reaction is measured as enzymes act as biological catalysts and the concentration of reactants change rapidly. The temperature coefficient is used to quantify the effect of changing temperature. This shows the change in rate of reaction caused by 10-degree increase in temperature and is calculated by higher temperature divided by lower temperature. The temperature must differ by 10 degrees only. Do not forget that. The following practical will be measuring rate of reaction by measuring the volume of oxygen gas produced to calculate the rate of reaction. So what do you need for the experiment? You need a water bath, boiling tube, bung, soaked peas, hydrogen peroxide solution, delivery tube, gas syringe, stop clock, and mortar and pestle. So grind the measured mass of peas in distilled water and place in a boiling tube. Add 5 cm of hydrogen peroxide solution into the peas. Fit the syringe into a delivery tube and the delivery tube into the boiling tube with a bung. Place the boiling tube into a water bath at a known temperature, so this would be your control variable. Measure the volume of gas every 30 seconds for 5 minutes and repeat the experiment at different temperatures and plot a graph of temperature against ethanol concentration. So your temperature is going to be your independent variable and the rate of reaction is going to be your dependent variable. So what are the different risk assessments? So you need to acknowledge the biohazard, which is the green peas. So possibility of contamination can take place. So use disinfectant and wash hands with soap after handling. There is the risk of broken glass, possibility of cuts from sharp edges. So it is safe to keep the glassware away from the edge of desks. In case of an accident, elevate the cuts, apply pressure, but do not remove glass from the wound and seek further medical assistance. There's also the risk of hot liquids because you are dealing with different temperatures. So risk of burning skin is possible. So handle hot liquids with care and use tongs to remove boiling tubes. In case of burns, run the burn under cold water. And finally, there is the hydrogen peroxide, which can irritate eyes and cuts. So use eye protection and avoid contact with skin. However, in the case of an emergency, wash the skin and eyes with cold water. So what can you conclude and what are the different things you can analyze? So the rate can be calculated by dividing the volume of gas produced by time. And the temperature coefficient can be calculated from the data collected and can only be used up to optimum temperature. So what the formula for the temperature coefficient would be dividing the higher temperature over the rate of the higher temperature over the rate of the lower temperature, and you will get a number which will tell you what the temperature coefficient is. So that is all from Professor SNAB today. Hope you found this topic useful. This is a core practical, so you should definitely know this. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.